Metabolism is a word used to describe all the chemical reactions inside our body that keep us alive. This includes digesting food and making essential building blocks such as fats, sugars, and amino acids that can be used to build muscle or burn energy. In other words, metabolism is essential for physical health. And when metabolism is altered, we get diseases like obesity, diabetes, and cancer. Our lab studies metabolic pathways in order to figure out what's going on in human diseases and how we can treat them. We ask questions like what cellular processes and pathways contribute to disease, and how do the cells acquire the nutrients to fuel these pathways. One disease we study is cancer. Cancer cells have altered metabolism that allows them to rapidly grow and proliferate. In order to keep expanding and growing, cancer cells take up large amounts of nutrients for fuel. We study how these cancer cells acquire so much fuel so that hopefully we can block that pathway and identify a new cancer treatment. To study these metabolic pathways, we trace molecules on their journey through the body. Instead of people, we use zebrafish, mice, and cells to represent human bodies and disease. To trace the molecules, we use a special process called isotopic labeling. Isotopes are chemically identical molecules with a special tag. We use that special tag to track the journey of molecules after we inject them into the bloodstream of animals. After being injected, the molecules go down specific pathways and integrate into the body. Once the isotopes are integrated, we collect samples from the animals and look to see where the isotopes ended up. This information acts like pieces of a puzzle that we can put together to figure out the whole journey of the molecule. To figure out where the molecules ended up, we grind up different tissue samples and run them through a series of processes to separate them into different groups. When we have just the groups of molecules we want, we put the sample in a machine that separates and identifies the molecules in the sample. This is called liquid chromatography mass spectrometry. LC-MS gives us peaks that help us see where a special label ended up. We then use this information to look for areas where there's a difference between the healthy samples and the disease samples. This cues us to which metabolic pathways are relevant and how the pathways are altered in disease. An example of this approach in action is the study of liver cancer in a zebrafish model. We injected fish with isotopes and discovered that the fish with tumors had more of a certain kind of lipid species, and we used isotopes to prove that the lipids were being made by a pathway that could be targeted to block cancer growth.